nurture condition.
Anyone? Psychologists?
Sure, I'll jump in. The standard answer is it's a combination of both. So, yeah, when we look at processing spatial relationships and then, on the other hand, processing verbal relationships, there are studies that suggest that women are better at verbal processing and men at spatial processing. So whether that's inborn or not, I'm not sure about that, but the phenomenon exists and the cause of it is probably a combination of both things. I don't know if anybody else wants to jump in on that.
Well, there are certainly gender differences that have been seen. I suspect that they've changed with age and that there's a convergence as we get older. But people, now the NIH requires that we actually do experiments with males and females. So things are getting better. Any, okay, wait, back there. One of you,
just to follow up on that question, Stephen Ceci at Cornell did an interesting study on this male-female difference in wayfinding. There are tunnels underneath Cornell because the weather's so lousy. And he took male and females and said, let's try to find your way from the student union to the library. And what he did was he systematically maneuvered the arrows or the signs and asked people to do this. And females were very much compliant with going with where the arrows pointed, despite that they may not be pointing in the right direction. And males were quite resistant, saying, it's this way, I don't care what the sign says. And so there may be something to follow up on the panelist comment about how, if I have a spatial dead reckoning sense, I tend to trust it, but if I'm particularly verbally skilled at mediating my world, arrows and landmarks and those sorts of things are very useful to me. Which is why, like, when my wife says, let's get directions, and someone says, go southeast about 1/4 mile, she says, what does that mean? That means go to the post office and make a left. That's useful, whereas the other one is not quite so useful. So there does seem to be some sort of strategic difference that may be related to brain, but it's probably moderated by a lot of environmental factors beside that.
great, thank you. The other?
Hi, I'm Susan Mag-Salmon with the Brain Science Institute at Johns Hopkins. I had 2 questions. One is around the medial temporal lobe memory. Do you have any idea how many maps we lay down sort of over our lives?
Depends on how long we live.
I think this is probably a better question for Michael and Beeb. But I think many computational models have tried to predict how many arbitrary associations could be formed in hippocampus and medial temporal lobe depending on the number of neurons there and the number of synaptic connections that they make. And I think that the only comment I could say, and these are all computational models because we can't actually measure that or probe all of those dimensions experimentally. But I think the interesting point to show is that there are different ways in which these networks at subsurb memory can form differences for experiences. And having multiple ways that an event can be distinctly encoded, either it's in 2 different maps, or you have a map that's changing because of the activity patterns of the neurons that are representing that map. All of those differences can increase the number of maps that can be stored and the number of ways in which that network can form discrete representations. So I don't have a number for you.
Well, and it leads to my second question, which is really around developmental work. My work is with education children, babies through 7 years old, and I was really interested about the way babies do this and young children do create these maps, and if there is a progression of more maps early, very early developmentally, and if you've looked at this in humans at all to see this.
So I can only speak towards animal models, and that's the best way to address neural network actual mechanisms because we can go in and manipulate and assess things you can't do in humans. But in a rodent, in which the circuitry and the anatomy is homologous to ours. That we have looked at the development of these different kinds of cell types when they come in line and how they map out space during the course of development. And actually, when the first time a little rat pup, as big as it can, to stick some electrodes in their head before their eyes are open, weans from their mother, they immediately have hippocampal place cells, they immediately have head direction cells, and border cells in the medial interneural cortex. However, it takes a long time for rats to develop grid cells. They're the last cells to come online. And so there is spatial information coding in these young organisms as they just begin to start navigating away from their mother and and exploring space. But those networks and those cell types continually develop over time, and they do not reach the levels of adulthood after some months afterwards.
It might also be interesting to think about this overlap between caudate nucleus and hippocampus. It may be early on there's a habit learning system mediated by the caudate nucleus that kind of kicks in, and it probably takes a while for the development of the hippocampus to a point where you're forming memories. So,
Could you pass the... I'll get to you, Tom.
Hi, this is a question for David Salmon. I was wondering if there were any correlations with regard to the different people, dementia, Parkinson's, and Alzheimer's, to correlate them back to their original abilities. So example would be an architect who had more spatial ability versus a poet who has a different, and how they would respond to those different tests.
yeah, that's a good question, and it's something we thought about. It's very difficult to do those kinds of studies, but anecdotally, I would say that there's not. In fact, it's almost the other way around. It seems like we've seen a number of architects, former architects, and engineers who developed Alzheimer's disease and actually showed their greatest deficit in visuospatial abilities. And we're not sure why, or if that's just anecdotal and chance that it turned out that way. But it's not like a lifetime of performing a certain kind of task is protecting you from the ravages of Alzheimer's disease on whatever brain area is mediating that ability. So, that's just an anecdote. But,
Nothing imprinted.
not that I know of offhand. Yeah. I'll think about that, though.
We actually have a question back here. She's been waiting for us.
Thank you.
I wanted to come back to the male-female spatial cognition question. I had a look at that literature to look at the exact experiments that were proposed to demonstrate this male-female difference. Many of the ones that demonstrated it were with the standard cognitive tests that you might find on an SAT, where you have a three-dimensional drawing of a block, etc. And they were showing differences there. However, when they looked at experiments of more ecological tests of natural wayfinding in built environments, those differences were lost. I think most interesting to the applied audience, I have a history with Gensler, who became a very large firm, and one of the reasons they became a very large firm is they realized the value of looking inside of the architecture and brought in the profession, who at that time, a zillion years ago, happened to be females working in interior architecture and interior design, and realized that the combination of both of those perceptions was what made the entire building work well for the people using it.
Thank you. Another question? This lady, and then Tom. So,
I'm wondering, in any of these studies, I guess you could do it with a rodent or a person, is there any time where the animal or the person is moved as opposed to moving themselves through the environment? Like if you take the rat and kind of, put it on a little skateboard and move it around, does it have the same, do the cells fire the same way? Or if And I'm thinking about the phenomenon of, my name is Alyssa, I'm an architect at the Portico Group in Seattle. We do a lot of work with spaces for kids and families. And there's a lot of discussion about kids exploring their own environment versus being moved around in containers of various kinds. And so I'm wondering if that happens, if that development happens differently or sooner, if there's anything that relates to that.
There was one study done in the 90s by Bruce McNaughton, and that was at first I was amazed they could even train rats to do this. But they either had rats running around a track exploring their space, as I've explained, or they trained them to sit on a little car with a lever and put it forward and back to brake or go forward. And so they did the same kind of recordings to look at these place fields to see if the spatial dimension or the firing properties of the cells were different if they were running around and exploring, or whether or not they were moving through the space without their own self-motion. And so there are differences in the spatial tuning of the cells. Space is represented, but the acuity, the finiteness of the space or representation is a little weaker when they are not actually motivating. It sharpens up the spatial representation, and it develops a bit faster if you're moving through space and are a rat. So I don't know of similar experiments. People are doing similar experiments now with virtual navigation, rats that are head fixed, and they're going sitting head fixed because now people are imaging dendrites and synaptic connection between cells and seeing how they change over time. And the animals have to be head fixed under a microscope, but now they're moving through space visually in these virtual reality, and they're looking at whether or not the cell types are changing then. They still have spatial tuning. Again, it's not as sharp, but they're doing virtual reality and as humans as well, looking at larger kind of activity patterns. But I would say there are slight differences over in when they've looked in rats.
Okay, Tom? And then
there's a great children's story about a mouse that drives a little car around, which I remember very clearly. I just had a comment that's particularly relevant to David's talk, which is, generally speaking, there's utility and value associated with locations in space, and this is true whether they're familiar spaces and, to a lesser degree, unfamiliar spaces. And I think one of the things that goes wrong in dementia is the loss of that association of utility and value with space. And there's a member of our board, John Zeisel, who has developed techniques for trying to overcome that limitation in people with Alzheimer's dementia. And generally speaking, the type of things that he does, he runs a series of Alzheimer's facilities on the East Coast. So what he does is put clear identifiers, explicit identifiers of the utility and value associated with different locations in the space that those people are living. So for example, you come out of your room and you look down one end of the hallway and there's a fireplace, and that's an explicit way of identifying that the utility of that space is that it's a sitting, and the value is it's a sitting room, and the other direction there is a bookcase. Yeah. So the utility and value associated with that location is it's the library, and so on. So these may be ways, I just mentioned them because there is actually a literature on this, and there are efforts to do this to try and overcome that loss of the association between the utility and value and the location in space.
I'd like to make a comment. As we understand better which parts of the brain are involved and what strategies are involved in those pathways, as we've heard today, then you can design some remedial or some training paradigms that are appropriate. If we think that everybody's allocentric, we probably miss a lot of things. So it's additional knowledge coming from the neuroscientists, I think, can be very illuminating. There was another, you have a question?
I just wanted to ask about the grid cell map. Have you determined what causes sort of the first grid cell hotspot in a given map, and are the maps continuous? If you take the rat and put it into the next box, does the grid continue into the next box, or is it a discrete map there?
So the pattern of the grid is internal. So the spacing between the nodes and the actual grid itself does not change with any kind of external stimuli or anything you do. That's established between the cells. Cells neighboring each other will have the same spacing within their grids. And then there's kind of modules. There's modules in the brain everywhere in your brain where you're storing discrete steps or visual scenes. But here in the grid cell system, you will have a module where all the cells have similar spacing, and all of a sudden you'll jump to another module where they have different spacing. Again, their nodes are in different places on the unitary grid. So that, I think, was one question. Then your another part of your question was to say, is this map just continue? Where does it stop? If you were to move somewhere else in space, would you just be in a continuum of that spatial map, or does it change? And the question would be, is the animal aware that they're in the same space, right? If you have two boxes together, and the animal was trained to explore those two boxes in two different places by having a corridor between the two, and he ran around, then went over to the other, ran around, and they're connected, there'll be one continuous grid of that space if you move the walls and sampled the cell. If you train the animal in the exact same positions without the corridor, you have two independent spatial maps of those environments. So, but the animal, is a small, the walls are, they're about 50 centimeters high. That's their main perception is of those walls and maybe some of the distal cues. But there's a lot of work looking at what kind of cues are needed to establish and generate new independent maps versus bind them to maps that are already formed. And that depends on the memory system you're recording from, the hippocampus or entorhinal cortex, and how the animal learns or learns a novel environment from the beginning. And we can kind of modify the dynamics of remapping these changing maps and things in the circuit by modifying how the animal originally learns about a certain space. So that's kind of dynamic.
Okay, one last question. Anybody? Down here.
Continuation of that question. Is there a difference between if you then put the corridor, once they learn separately in each individual box, is there a correlation? Do they recognize the second box
as one of the initial?
So here's an
maps.
experiment we did, and the question is, once they've learned without the corridor, do they keep those distinct environments? And here's an experiment that we asked to kind of address that question. So we trained an animal in two different boxes, and they had two distinct maps for those boxes. Then we slowly moved them together into the exact same space, and we wanted to know if the cells would then represent that space or the space that they had associated to that environment. And when we moved them into the same location, the animal continued to have distinct spatial maps for the same place because he learned them in two different locations. He had to be trained with the corridor to know there were two places. Then we took the corridor away, and then we maintained those representations for some time. With time, as the animal realized he's in the same place and started taking in additional cues about the environment that we tried to impoverish as much as we could, the network did reorganize at some point and a new map formed for that space.
Okay, I'd like to thank the speakers, and now it's time for you to use